And last but not least on our little circle here through biotechnology are genomics. Genomics is the study of a complete set of genes from a human genome and their interaction. We have many prokaryotic and eukaryotic genomes sequenced. And why do we actually do this? Evolutionary relationships, we can compare organisms to one another, and then actually interpreting the human genome, figuring out what genes actually do. So this is just an example of some completed genomes. The first completed one was in 1995. Okay, it was a form of bacteria. Saccharomyces cerevisiae in 96. That, when we talk about doing genetic studies in yeast, that is the strain of yeast that we are using. And yes, it does make a difference. Ask any brewmaster. Yeast makes a difference. E. coli in 97. 98 was C. elegans, which is a nematode worm. It is a model organism for genetics. In 2000, we hit both Drosophila melanogaster and the mustard plant, which were biggies. 01 was mice, 02 was rice, and then in 03 was us. Note the number of genes. It was a real kick in the seat when we figured out that we had fewer genes than rice did. But if you go back to the previous unit and are talking about alternate slicing and being able to put exons together in different ways, we're simply more efficient than rice. That's what we tell ourselves, at least. Helps dampen the blow a little bit. Human Genome Project. It began in 1990, and it was this incredible undertaking. The goal was to sequence the entire human genome, all 3.2 billion base pairs, 3.5 billion base pairs. There's somewhere around in there. It started as a government project. Then there was a company, Celera, that came along and started competing against the government, which was the best thing for it because the government is not always the epitome of efficiency. They ended up actually joining forces in 2002. The original paper came out saying the sequencing of the human genome. I was in college. My professor walked in. I was taking cellular biology at the time. My professor walked in and was like, "Welcome to the post-genomic era." It was pretty cool. Okay, I know, dorky Mrs. Morgan cool, but cool nonetheless. By 04, it was 99% completed with 99.99% accuracy. The goal was to determine the nucleotide sequence of all human DNA and then identify and sequence every gene. And then we wanted to get genetic and physical maps of the chromosomes. Now, we actually used a composite reference of genomes of multiple different individuals. You wanted to have variation in ethnicities, and even, like, as I recall, there might even be some from tribes in the Amazon rainforest and things like that. Like, we really tried to get a wide collection of the genetics of the human race. Genome consists of 3.2 billion base pairs, somewhere between 21,000 and 26,000 genes. So we know that only 1.5% of our DNA codes for proteins, transfer RNAs, and ribosomal RNAs. 24% codes for introns, which we talked about. That's what gets removed when we're putting together messenger RNA, and regulatory sequences, switches, things like that help turn genes on and off, and we'll talk about that later. 15% is unique non-coding DNA. This is where your STRs come into play. 15% is repetitive DNA unrelated to transposable elements, and then 44 is related to transposable elements, which are jumping genes. I'm not going to get into jumping genes with you guys right now, but it's a pretty interesting topic. The whole genome shotgun method, okay, it was developed in 92, and this is actually what determined the entire genome. Used to be in sequencing a genome, you formed a linkage map. We looked at linkage the last unit, and it was how close, or two sections ago, how far away are genes from each other on a chromosome. Remember, if they're close enough together, they always get inherited together. Okay, they're linked. So we looked at the pedigree analysis containing more than 5,000 genetic markers. You then constructed a physical map, and then you started determining the nucleotide sequence. The shotgun method, we skipped steps 1 and 2, and we started directly with nucleotide sequencing. I had the privilege of having Mark Adams, who developed this technique, actually on my thesis committee. He was a really nice guy, very down to earth, given that he's sort of a genetics god. And he did this. He's a brilliant geneticist. He also happens to be a brilliant computer programmer. And he developed the computer program that actually allowed this to happen. So I cut DNA samples with restriction enzymes. I clone the fragments. And then what you do is you go through and you sequence, which is, you can figure out where A, T, C's, and G's are. Like, that's not a difficult thing to do. And then the computer program went through and found overlapping sequences and started putting the DNA, then the sequence, actually in order. And this made this all process happen literally tens of thousands times faster. When I was freshman in college, they expected the human genome to be like completed by something like 2010, and it ended up being in 2003 is when they call it officially completed. Now, genomics blew the door open on a lot of things, especially ornithology. Birds were a big one. We sort of thought birds were related genetically, evolutionarily to one another, based on ecological niches and based on like wing structure, beak structure, things like that. Then we started looking at the genomics, and we realized that some birds that we thought were closely related were not remotely closely related genetically. Then you take the next step and it goes into proteomics, which is looking at the full set of proteins produced by organisms. This picture here, myosin and actin are both proteins that are involved in muscle formation. Okay, myosin and actin work together to help constrict and to relax your muscles. And you can see here it's a bunch of different type of fish that you're actually comparing them between. Now, why go to proteins? Well, genetically, we know that just because I have an alteration in the gene or an alteration in the DNA sequence does not mean that I even have a change in the amino acid sequence, right? Because of that wobble position at number 3 in a codon, you could have a change and your amino acid might not even be different. Or you can have one that is so minor that it really doesn't impact the shape or the structure of that protein, the tertiary structure, and therefore it does not impact its functionality. So proteomics is then beginning to go through and look and see where do our proteins actually differ from one another, because then you're going to see some pretty big evolutionary steps. And that can help us go through and look here then at our evolutionary tree. You know, this is fish. And this is based then on proteomics, being able to go through and actually look at the proteins. This is a nice little video on it. Okay, and that is the end of our lectures on biotechnology.